Okay, so let's get started. So welcome everyone to today's ImmuZoom seminar. So it's really my pleasure to welcome our today's speaker, Dr. Shuo Wang, from Insitute of Microbiology, Chinese Academy of Science. So Shuo currently is a principal investigator in key laboratory of pathogen microbiology and immunology in Institute of Microbiology, Chinese Academy of Science. So a very, very brief introduction, since I can't find so much information about Shuo's on Shuo's website. But anyway, so I think Shuo did his her undergraduate study in Zhejiang University in major in bioengineering in School of Materials and Chemical Engineering, which I think is it's a quite good start if you want to study biology, because I think it's a combination. Now it become a trend to develop multi-discipline projects. So after she completed her undergraduate study, she decided to pursue her PhD training in Institute of Biophysics in Chinese Academy of Science in biochemistry and molecular biology. So after she completed her graduate study, she actually started as assistant researcher in Institute of Biophysics until I think 2015, she became a independent researcher still in Institute of Biophysics in Chinese Academy of Science. I think from 2019, she moved to current institute, which is Institute of Microbiology. Actually started her a new lab as a new research program. So Shuo's lab and mainly interested in mucosal immune system, respiratory pathogen and immune responses, mainly focused in studying innate lymphoid cells, inflammation, and also colon cancer. Her lab actually did a lot of work in intestinal mucosal immune response regarding to lymphocytes and lymphocytes phenotype, and also intestinal disease development and regulation, and particular focusing how the interaction between the intestinal commensal bacterial and then the immune system and how they regulate the disease development, which I think we're probably going to hear a lot today. So I think without further ado, thank you so much, Shuo, to contribute to our seminar series. So we are looking forward to your talk. Please go ahead.
Thank you, Chen. Thank you for your invitation and the introduction. I'm very happy to be here, and I think it is really a wonderful seminar series for the immunologists to share their work and ideas. And as the Chen's introduction, our group major work on the mucosal immune system. And today I will introduce you something about the tissue-resident commensal bacterial and the tumor immune surveillance. Our group major work on the mucosal immune system. So first, I would like to say something about the interaction between the mucosal immune system and the microbes. You may say that the mucosal immune system compromises the largest part of the body's immune tissues, including the respiratory tract and the gastrointestinal tract, etc. And they are exposed continuously to the antigens and the materials from the environment. So when meet the pathogens, the innate immune cells are activated, and they capture the pathogens to the lymph node to activate the T and B cells. And the T and B cells traffic back to the infected site and contribute to the antigen-specific response. Then The mucosal surface also meets a large amount of commensal bacterial. The interaction and the crosstalk between the commensal bacterial and the mucosal immune system may happen every minute. Under physical conditions, the microbiota will help the development of immune cells. So they may play a fundamental role in regulating and training of the immune system. So the commensal bacterial may be beneficial for the immunotherapy, vaccination, and the control of infection. Also, the dysbiosis of the microbiota will contribute to the autoimmune diseases, energy, and the metabolic syndromes. As I mentioned, the gastrointestinal tract has a lot of commensal bacterial. It is also the major source of the commensal bacterial. The aerobic species are major in the upper small intestine, and the anaerobic bacterial dominates in the colon. The dysbiosis of the microbiota has been associated with many different types of diseases, including infection, colitis, and cancer, especially the colon cancer. Then we move to the colon cancer. Colon cancer is also called a colorectal carcinoma, is one of the most common malignancies in the world. A lot of work has showed that the microbiota influences the intestinal tumorigenesis through several mechanisms. The Fusobacterial, let me see the one, Fusobacterial had an antigen FADA can bind the E-cadherin on the intestinal epithelial cell, that is IECs, to activate the beta-catenin pathway. FN also has a pro-inflammatory effect on tumor cells. Furthermore, as reported by Fang Jinruan's group, FN also has shown to modulate the autophagy in IECs. Then you may see that the E. coli strain covers the PKS island and encoding the genotoxin colibactin, which are frequently observed in human colon cancer. And the ETBF strain also causes inflammation and tumor induced by the ROS and the DNA damage. Also, some bacteria that produce hydrogen sulfate and also promote the inflammation. You may see that this bacteria can drive or promote the tumor progression by release some small molecules or by affect the epithelial cell of the intestine. Also, during the discovery of the tumor environment, a lot of work showed that the tumor environment has microbes that also exert the local effect. The history of the finding of the bacteria in tumor can back to as early as many thousand years ago, when Egypt physicians treated tumor by incising swellings and then cause infection. In the 13th century, a person had a huge growth of tumor on his leg, developed a severe infection after amputation, but the cancer never returned, indicating that the infection may influence the tumor development. In the 18th century, William Coley successfully treated the sarcoma patient with a vaccine made by two inactivated bacteria by direct injection into the tumor site. In the 1911, Peter Rose discovered that the breast cancer infiltrate of chicken can lead to a transmissible tumor. This may be caused by the parasite organism. In the following decades, people have discovered a lot of viruses that can induce the tumor, the cancer, the tumor progression, such as EBV, HBV, HCV, and HPV. In the 1983, Marshall and Warren successfully cultured HP, and they found that its role in peptic ulcers, and also found this bacteria can cause stomach tumor. With the development of sequencing technology, more and more articles have reported the existence of bacteria in the tumor environment and influence the tumor progression. In the 2020, Ravid Strassman's team conducted the first comprehensive analysis of 7 tumor microbiomes and provide intratumor spatial distribution of this microbiota. They also raise a lot of questions about the intratumor bacteria, including their translocation, viability, communication, metabolic activity, and pathogenesis, and so on. This is a long story about the intratumor bacteria, and we then look at the tumor environment. You may see that the tumor environment constitute a lot of not only tumor cells, but also the immune cells, blood vessels, and the stromal cells, also, of course, the bacteria. All this constitute the tumor environment and play a very important role in regulating the development of tumors. Just like the old saying, the Rome wasn't built in a day. The cellular transformation and the tumor development also result from an accumulation of mutation and epigenetic changes. At the same time, our immune system also changes. First, they protect the host from cancer development at early stage by activating the NK cells or CD8 T cells, which called the immune surveillance. But during the progression of the tumor and the microenvironment also changed, the tumor cells may downregulate the expression of MHC molecules, and the immune system also be suppressed by the cytokines such as IL-10 and TGF-beta. The immune system become exhaustion or undergo metabolic reprogramming. This process is called immunoediting, which is showed by the Mark Smith group. And we also want to know whether the bacteria participate in the immunoediting process, and what's the composition of tissue resident, but not the tumor resident bacteria, and what kind of function do they have. We're also back to the colorectal cancer. We took this as a detailed analysis. We focused on the colorectal cancer for further analysis. We explore the role of intra-tissue microbiome in colon tissue in colon cancer patients. We collected the normal peritumor and the tumor tissues from each patient. Pay attention, the 3 kinds of tissue were from one person. That means they from the same person. 3 kinds of tissues from colon cancer patients, and we treated them and for the bacterial ribosome RNA sequencing. Since the tissue were from the same patients, according to the experimental design, we are able to exclude the individual differences and identify the bacteria that protect against or promote the tumor progression. You may ask a question, why you say that they may protect from the tumor progression, we think that the bacterial in normal tissue may have a protective role because, as I showed, the immune system in the normal tissue may have the immune surveillance function. So we think the bacteria in the normal tissue may have also have some effect role on the immune system of the normal tissue. So we hypothesize that this bacteria may have some protective roles. According to our data, we found that the normal and paratumor tissues display high diversity of bacteria. We also use buffer control to exclude the contamination. To exclude the individual variation of each patient, we compared the composition of normal paratumor and tumor colon tissues from each patient. You may see that the bacteria belong to the Bacillus family, that Ruminococcus, Doria, and Blautier were significantly enriched in normal and paratumor tissues. The correlation analysis also showed that the Lacinospiraceae family bacterial, Ruminococcus gonorrhoeae, Doria, and Blautier were the leading bacteria in the microbiome community of normal and paratumor tissues. However, the Fusobacteria was a core bacteria in tumor tissues. Therefore, we found that the bacteria belong to the Lacinospiraceae family, were the major bacteria in the normal and paratumor tissues. Then we want to investigate whether the intratumor bacteria, such as the RG and Ruminococcus gonorrhoeae and Blautiers, have some functions to regulate the tumorigenesis. We first use subcutaneous tumor models to test whether this bacteria affect the tumor environment. We also conducted the metagenomics analysis to analyze the bacterial species in the tumor. We also find that the Ruminococcus gonorrhoeae, we call the RG, and the Blautier products, we call the BP, were significantly enriched in the normal tissue. And so we use RG and the BP for further validation. We also use the Fusobacterial nucleotome and the Peptostreptococcus anaerobis, PA, which were reported to promote tumor progression as tumor facilitators. You may see that injection of RG and BP into the tumor can significantly reduce the tumor size in the wild-type mice, which has a whole immune system. Moreover, the tumor growth promoted by the addition of FN and PA can be inhibited by the addition of RG and BP. Also indicates the tumor suppression role of RG and BP. We also found that during our experiment, we found a small fraction of mice after the administration of RG and BP, the tumor even can be vanished, and also suggests the tumor suppression function of this bacteria. We also took advantage of the inflammatory induced colon cancer model, that is AOMDSS model. You may find when we oral gavage with this bacterial to mouse, the tumor size, tumor number were also significantly reduced by the oral gavage of RG and BP. Therefore, in this slide, you may find that RG and BP can inhibit the colon cancer tumor progression, both in subcutaneous and spontaneous tumor models. Then we inhibit the viability of RG and BP by heat killing. And we found that after the inactivation of RG and BP, the tumor suppression function of them were abrogated, indicated that the tumor suppressive function may be dependent on their viability. We also use noodle mice to analyze the function of the immune system. You may find that after the depletion of the immune system, especially the T cells, the function of RG and BP also abrogated. So this indicated that the RG and BP play an important role in tumor suppression dependent on the T cells, because the noodle mice do not have T cells. Then we further analyze the immune cells in the tumors. We have analyzed a lot of immune cells, especially CD8 and CD4 T cells. We only find that the tumor infiltrating CD8 T cells were apparently increased after the injection of RG and BP. We also find that the colonization of RG and BP in the tumor environment do not affect the addition of FN or PA. We also obtained the similar result from the AOMDSS model. Then we analyze the correlation between CD8 T cells and the abundance of RG and BP. You may see that the frequency of CD8 T cells shows strong correlation with RG and BP. This is from the patient's data. Therefore, we conclude that the tumor tissues which have high levels of RG and BP show more CD8 T cells. So RG and BP may positively correlated with the CD8 T cells. Next, when we want to know the transcription change of the CD8 T cells by the addition of RG and BP, so we did the transcriptional sequencing of CD8 T cells after the administration of RG and BP. You may see that some molecules such as granzyme and porphyrin and interferon which contribute to the immunosurveillance function of CD8 T cells were significantly increased or enriched after the treatment of RG and BP. We also found that in the CD8 T cells, their metabolism pathway also changed. You may see that the carbohydrate metabolism that is in green color or increased after the addition of RG and BP, but some pathway related to the lipid metabolism in red, that is in red color, was decreased after the RG and BP injection. So this data indicated that the treatment with RG and BP can influence the immunosurveillance function of CD8 T cells. They also affect the metabolism pathway of CD8 T cells. Then we want to know the function change after the treatment of RG and BP. We further confirmed the increased expression level of granzyme B and interferon gamma after the treatment of RG and BP. We also found that the injection of RG and BP promotes the cytotoxic function of CD8 T cells. Then we also deplete the CD8 T cells by antibodies. you may see that after the depletion of CD8 T cells by the anti-CD8 antibody, the tumor suppression function of RG and BP was abrogated. So this data also suggests that the Lactobacillus family bacteria RG and BP can promote the CD8 T cells activity, and their function of tumor suppression also dependent on the CD8 T cell. So we next want to know, where do the bacteria locate in the tissue? We next analyze the localization of RG in tissues by IHC analysis. You may say, according to our data, we found that RG could locate in the tissues, both in subcutaneous model and AOM-DSS model, by both intratumor injection or oral garbage. You may also see that RG that is in red color do not co-localize with CD8 T cells that in green color, indicate that the bacterial RG in the tumor may function by modifying the tumor environment, but not directly target the CD8 T cells. Then we want to further know the metabolic changes in the tumor environment after the treatment with this bacterial. So we extract the tumor interstitial fluid from the tumor, and for the metabolic mix and analysis. You may see that the addition of RG and BP apparently changes the component of the metabolites in the tumor environment. Many metabolites were up or down regulated after the RG-BP treatment. We figure out the enriched metabolism pathways shared by the RG and BP treated groups. You may see that there were 44 metabolites were upregulated, and 127 metabolites were downregulated in all the RG and BP treated groups. Among them, we found that the carboxylic acid and fatty acids groups were apparently enriched after the RG and BP treatment. However, we did not see apparent enrichment of short fatty acid after the treatment of RG and BP. That may be RG and BP may not change the short fatty acid in the microenvironment of the tissues. Importantly, we also find the lipids that called glycophospholipids were significantly reduced after the injection of RG and BP, indicating that RG, BP, or BP might affect the level of these glycophospholipids in the tumor environment. Then we analyze the metabolites in detail. According to our finding, the group of glycophospholipids were one of the major metabolite classes that were decreased along with the addition of the RG and BP. We found that this phospholipids increased with the addition of PA, but decreased after the addition of RG and BP. This also suggests that this Lactobacillus family bacteria, RG and BP, can change the tumor metabolic environment by decrease the level of glycophospholipids. So what's the function of these lipids in the tumor environment? We analyze the function in detail. First, we want to know whether these lipids can affect the growth or proliferation of tumor cells. We use different kinds of these phospholipids. That's a lot of kinds of belong to them. We use the major 5 kinds of these phospholipids to treat the tumor cell. We found after the treatment with these glycophospholipids, the tumor cell did not have a significant change in their proliferation or migration. So next, we analyze the effect of this glycophospholipids on CD8 T cells. You may find that these phospholipids can inhibit the proliferation and function of CD8 T cells. Also, we also use the tumor. We also use the subcutaneous mouse model to analyze the function of these lipids. We found that after the injection of this lipid into the tumor infiltrating CD8 T cells were significantly decreased, and the production of granzyme and interferon gamma were also suppressed, indicating their inhibition of the function of CD8 T cells. We also analyze the cytotoxicity of CD8 T cells. Function of CD8 T cells were also found inhibited by the addition of these glycophospholipids. Therefore, we think that the glycophospholipids inhibit the CD8 T cells, but not to promote the proliferation or migration of tumor cells. Then we back to this bacteria. We also want to know how this bacteria affect the level of these phospholipids. We investigate the we analyze the gene of this bacteria. We found that the gene of glycophospholipid metabolism pathway were significantly enriched in RG and BP. We also use the metagenomics analysis. We found that the intra-tissue bacteria showed a high level of PLDB and PLSC, which are 2 important enzyme participate in the degradation of these phospholipids. This data also suggested that the RG and BP might decrease the phospholipids by degradation of this lipid, because they have some important enzyme to degrade these phospholipids. Well, you may say that this enzyme enriched in also not only in the RG and BP, and when we analyze the metabolism of CD8 T cells, we found the metabolism of these phospholipids also, the degradation pathway also increased after the addition of this bacteria in the subcutaneous model. Then we want to know whether this bacteria can directly degrade these lipids. We use an in vitro assay. We cultured this bacteria with this glycophospholipids in vitro. We found that the glycophospholipids, LYSOPA and LYSOPC and this and PE, we found that these two kinds of bacteria can directly degrade the lyso-PC with a high efficiency. When we inactivate this bacteria by heat killing, their degradation role was abrogated, indicating that the degradation of phospholipid depended on their viability. Then we also use the mouse model to analyze the function of RG and BP in degradation of these lipids. We found that the phospholipids could promote, you may say, can promote the growth of tumors in a subcutaneous mouse model. After the injection of RG and BP can significantly reduce the tumor growth in the mouse, and we also then further analyze the function of CD8 T cells. You may also say that after the addition of this lipid, the function of CD8 T cells were suppressed. But then when we also inject the RG and BP into the tumor, you may see that the function of CD8 T cells can be restored after the addition of this bacteria. Therefore, we conclude that after the addition of RG and BP, the function of CD8 T cells were restored. And we then finally we also validate this finding in human patients, in human samples. We also find that this phospholipid were enriched in tumor tissues, and the higher frequency of CD8 T cells and the lower level of this lipid were observed in the RG and BP high group. Therefore, we conclude that this Lichenospiraceae family bacteria, RG and BP, can degrade the extracellular glycophospholipids and restore the immunosurveillance function of CD8 T cells. That's a whole story about the intra-tissue bacterial function. To conclude this part, we found that the commensal bacteria belonging to the Lichenospiraceae family, including the Ruminococcus glandulus RG and the Blotier product BP, were enriched in the normal tissues. According to the immunosurveillance theory, this bacterium may have a protective role in promoting the immunosurveillance function of immune cells, and the tissue-resident RG and BP reduce the colon tumor growth and promote the activation of CD8 T cells. The intra-tissue RG and BP can degrade the phospholipid and maintain the immunosurveillance function of CD8 T cells. Therefore, the intra-tissue Lichenospiraceae family facilitates the immunosurveillance function of CD8 T cells and so then control the colon cancer progression. That's all for my talk about this short story, and I would like to thank my collaborators in the Institute of Microbiology, Chinese Academy of Sciences, and also some important collaborators such as Professor Chunjun Guo in Cornell University, and our sample from the patients were from the West China Hospital, and so on. We also thank all the invitation of Chuan and all the audience. Thank you very much. Happy to take questions.